Hello, hi. Today we are going to learn analytical method validation. So, in the analytical method validation, what are the parameters will be there to perform the analytical method validation? So, how will you perform analytical method validation? If someone asks you the question regarding analytical method validation, so you will come up with an answer as per our SOP. So, every individual company or organization having their own standard operating procedure. So, all the persons may tell we will perform analytical method validation as per our SOP. Most of the companies prepared SOP based on the reference to the guidelines. So, let us see the guidelines. What are the guidelines available? So, the first guideline is ICH guideline, International Council for Harmonization. And the second guideline is FDA CDER, Center for Drug Evaluation and Research Guideline. Some companies will take reference from Indian Pharmacopoeia IP guideline. Some companies will take reference from USP, United States of Pharmacopoeia, and some are taken from UNODC, United Nations Office on Drug and Crime. So, some companies will take ANVISA Brazil. This is a Russian drug corporation. These guidelines are available to prepare our SOP. So, let us check what are the parameters available in the guidelines. So, analytical method validation parameters comparison between above said guidelines. As per ICH guideline, there are some parameters available which are accuracy, precision, repeatability, intermediate precision, specificity, detection limit, quantification limit, linearity, range. These are the parameters in ICH. The next guideline, FDA. AMV as per FDA is the parameters given as specificity, linearity, accuracy, range, quantitation limit and detection limit, precision, repeatability, intermediate precision and reproducibility. These are the parameters given in FDA guideline. The next one, USP. As per USP, accuracy, precision, specificity, detection limit, quantitation limit, linearity, range and robustness. These parameters given in USP guideline. The next one, IP, Indian Pharmacopoeia guideline. As per Indian Pharmacopoeia, accuracy, precision, repeatability, intermediate precision, specificity, detection limit, quantitation limit, linearity, range and robustness. As per IP, these are the parameters. Next, as per UNODC. As per UNODC, specificity and selectivity, and the next parameter, limit of detection, and the other parameter, precision. Next parameter, linearity and working range, and accuracy within the laboratory, reproducibility and repeatability. Next parameter, recovery. Next parameter, uncertainty of measurement, and the last one is stability. These are the parameters given as per UNODC. Next, the final, AMV as per ANVISA Brazil guideline. This guideline given as specificity and selectivity, linearity, range, precision, dissection limit, quantitation limit, accuracy and robustness. These parameters are given as per ANVISA. So we will check all the guidelines at single time. So comparison of all these guidelines, ICH, FDA, USP, IP and UNODC, ANVISA. So as per ICH, these parameters are given. FDA is mentioned these parameters. As per USP, following are the parameters. IP given as these parameters. UNODC, these are the parameters given. And finally, ANVISA given these parameters. If you check these parameters, most of the parameters are similar to each other. However, one or two parameters or variation is there in these guidelines. Let us see the different parameters. In the USP, robustness is the different parameter given as axis, and IP robustness is the axis parameter, and stability is the axis parameter in UNODC, and robustness is the axis parameter for ANVISA. So why we are compare all these parameters? So if you verify these guidelines carefully, the order of the parameters are different from each other. In some guidelines, the first parameter given as accuracy, in other specificity, in some are accuracy. So order is changing. In the second one, some are in linearity, other one is precision, some are limit of detection. Like that, the order of parameters was different from all these guidelines. So do you understand why this comparison is required? So why you are comparing all these parameters? To know what are the parameters available in different guidelines, and which are the major, which are the primary parameter to perform the validation. So we will discuss in the next video how many parameters should be performed during method validation, and what is the order of the parameter should perform the validation to comply all these guidelines. Thank you very much. If you like this video, please subscribe this channel, like the video and hit the bell icon. Thank you very much.